So Larry, how did you first get interested in rhythms?
I went to the Institute of Animal Behavior that was run by Daniel Lehrman, who was studying ringdoves as a bird version of a laboratory rat. And there, in one of the classes that I took in my first year, we drew straws as to what topics we could get. And the topic that I wanted was on circadian rhythms, and I didn't get it. So I wrote a paper for this particular seminar class on the pineal gland, and this was in the early 1970s. And it turned out that that was the moment when Russell Reiter was first producing some of his really exciting work on the role of the pineal gland in regulating reproductive activity in golden hamsters. And there were 500 other publications about up to that point, and none of them meant anything until Russell Reiter produced this body of literature which grew extremely rapidly over the next decade. And it turned out that the pineal required a circadian clock for its proper regulation and seasonal interpretation of season. And so that got me very excited, this interplay between day length assessment and seasonality and reproduction.
So it came out of a classroom experience.
Yes, that plus my own interest in birds, that I've been a bird watcher all my life. So at the end of that time, at the end of the graduate school, where I did work on hormonal regulation of guinea pig sex behavior, I was looking around for some place to go do a postdoc, and my advisor in graduate school was a man named Harvey Fader, who had been a postdoc at the Oregon Primate Center, coincidentally with a guy named Irv Zucker, who was also a postdoc. And it turned out that Irv Zucker had published a whole bunch of papers as a postdoc on the hormonal regulation of guinea pig sex behavior, which I cited extremely heavily in my thesis. And so it came down to two choices: going to UCLA in the Brain Research Institute at UCLA, or going to the Bay Area and living in the Bay Area and working with Irv Zucker. Hey, no brainer. Go to Berkeley for 3 years.
And you did. I remember the first time I met you. It was at Versailles at the first European Gordon Conference. Do you remember that? We were sitting at a table. It was you and Ray Silver and myself. And we admitted to each other that I was an immigrant, but I was sure you weren't. We went around the table and all admitted we felt like immigrants to the field. Would you care to comment on that?
Well, that was true, because when I got to Berkeley, Ben Rusak was still there, and Ben was really the first person to push Irv in the direction of circadian rhythms. Fred Stefan had gone through the lab already, but just as an example of how naive the Zucker lab was, it was beginning to turn when I got there. But they had just published a paper. I think it was the first paper from the lab on circadian rhythms, and they plotted actograms with tau less than 24 hours sloped to the right and tau greater than 24 hours sloped to the left. And of course it's purely arbitrary as how you do the slope, but it's totally backward compared to the standard for the field. So that was the level of naivete that they had to progress from. And in addition, a bunch of Benji Rusak's data was missing when I got there, and had been missing for some time, and they had no idea where it was. And while I was there in the first few months after I got there, they found it embedded in the ice in the freezer of the laboratory refrigerator. We were outsiders. Irv came from outside of the field, and he always, I believe, felt that way, even though the field began to, at some point, coalesce around him.
Oh, absolutely. I remember that. So how do you think the field has been affected by immigrants?
Much for the better. I think, in some respects, one of the negative features of this field, and it's true for probably all fields, is inbreeding. where the same ideas are circulated and recirculated, and it's hard to break out of the mold. And the most recent example of immigrants that has had an enormous impact on the field is David Burson. And David Burson is a retina physiologist and anatomist, and it took him to demonstrate to the circadian rhythm field that there was a photoreceptor that was not a rod and not a cone that contributed mightily to the circadian rhythm regulation.
It seems in my following your research career that you have exploited the comparative approach very well. Would you care to comment on that?
I think that is probably truest with respect to appreciation of the role of rhythms in the regulation of reproduction. I won't say for myself particularly, because I didn't really work on some of these other hamster species, for example, the Turkish hamster or the Siberian hamsters, but folks like Bruce Goldman and Irv's Lab and Bud Ruby in particular more recently have used those species to very great effect to refine knowledge about circadian rhythm system and light regulation of annual reproductive rhythms. I do think this trend towards the use of mice for almost everything is extremely disheartening. Again, it's sort of a subject-related version of inbreeding, in almost a literal sense. There are some people doing fantastic work on other species who can't get funding because they're working on other species and they're not on mouse. Of course, that's not to say that the mice are poor subjects, as obviously they're not, and they have enormous benefits. But I personally feel that people should realize the real benefits that come from picking species for specific reasons and studying them very well for those same reasons.
I know you've given a lot of thought to the various, you might call them accessory nuclei of the visual system and circadian regulation. Would you care to sort of summarize your thoughts on?
Well, I think one thing, that's a good question, because that's near and dear to me. And I think one thing that is getting lost with along with the loss of emphasis on other species, there's and the increased utilization of mice to focus on the circadian clock. One of the things that is getting lost is the fact that the suprachiasmatic nucleus sits in a context of the whole visual system and feedback loops within the visual system itself. So yes, the retinohypothalamic tract projection to the SCN is of primary importance, but there are these accessory routes to the clock, most notably through the intergeniculate leaflet. But it turns out that the intergeniculate leaflet is connected to 100 other places in the brain, most of them bilaterally and most of them reciprocally. And so it makes it a very key player in not only the circadian rhythm system regulation, but in the organization of a lot of presumably visually related functions, such as rapid eye movements during sleep, because it's connected to all the nuclei of the brain that contribute to not only sleep regulation, rapid eye movements in particular, but the motor nuclei for oculomotor regulation, and to the vestibular system, which is also involved in sleep regulation and in oculomotor regulation, as well as to the hypocretin system, which is obviously connected to sleep regulation. So, yes, the breadth of the system as a whole has to be comprehended at some intellectual level, and it's not. I mean, within the IGL, for example, there is a division of cells. It appears that there are two distinct sets, even though they're distributed in a semi-random fashion relative to one another. There is a group that is involved in circadian rhythm regulation, those that project to the suprachiasmatic nucleus, but all the rest of them perhaps the large majority project to all these other places in the brain, and nobody has the slightest bit of understanding about what those projections do.
Is there some area you'd like to comment on?
I enjoy this meeting.
Good, good.
I enjoy the group of people at this meeting come from such different backgrounds and have such different intellectual bents, with some extreme cases, I suppose, epitomized by Evelyn Satinoff at one point.
Yes.
And Marty Zatz. And Irv Zucker. I wish he would come to this meeting more often, but it's been a great collection of people. We should turn to the Stony Brook part of this story.
Definitely.
So I arrived at Stony Brook in 1981, and already there was Bob Moore, and he was physically located 2 floors above me in this 19-story medical complex. And he was extremely generous and invited me to use his lab for any and all histology purposes. We had our own animal recording systems to work down in the animal facility, and rudimentary, I will say, histological capability at the time. So we wound up doing all our immunohistochemistry and nearly all our microscopy in his lab, and it was that was a fabulous time. There were a number of people who came into that lab and flowed between my lab and his. And at the time, Raul Aguilar Roblero was there. They weren't necessarily there all at the same time, but they overlapped. Vinny Casson, Michael Roberts, Joan Spey, his technician who moved all around even when he went to Pittsburgh, Stefan Royce, Shiggy Shibata, Laura Smale, Ralph Johnson, who were postdocs. They were situated mostly with me, but again, it was upstairs and downstairs doing all this work. George Newman was there. George was a faculty member in the Department of Neurology as a neurologist, but he made his mark in the rhythms world. And it was, I apologize to anybody who I might have forgotten at the moment, but it was a great time with a lot of intellectual exchange, and Bob and I published a number of, I think, quite respectable papers together, and it was a sad time when he decided to leave. And I just remember 2 more who made significant contributions from his lab in particular. Joe Riley, who actually preceded Bob to Stony Brook and set up the lab so everything was operational. And Joe was a very fine neuroanatomist who got his training in anatomy with Bob. I believe that's correct, but came to Stony Brook as a faculty member, and he did some very fine neuroanatomy on the SCN, but became seriously ill and completely debilitated, actually not long after I got there. And then Mary Bernstein, who was a research associate in the department also, and she did some of the good work, anatomical work on the development of the SCN. Unfortunately, she also got very ill a few years later. So I think that pretty much rounds out the group. Essentially, as I said, overlapping all the time. And as you say, it was an era.
It was an era, definitely.
Yes.
What do you, from your perspective of the research that you've done and the research you've seen evolve, look at as the major discoveries from your point of view?
The one that has had an enormous impact on the field, obviously, is it's sort of a general comment about the role of genes and molecules in the molecular clockworks, simply from a practical point of view, if not from a single pinpointed moment in time point of view, that's had a huge impact because it's generated so much work and so much productivity out of that effort. But if I had to pinpoint anything that was the single most important development, I would say, apart from the discovery of the retinohypothalamic tract, it was the discovery of the major cell group that contributes to the retinohypothalamic tract, namely the melanopsin containing retinal ganglion cells. I mean, that is such a big development, not only for our field, but it's likely to be of significance for the entire field of vision. And so it goes beyond the breadth of ours. And the third thing, actually, that is related to the first, the molecular one, is the extension of the entire clockworks mechanism to the periphery. And that is as important for the same reasons as the discovery of the melanopsin-containing ganglion cells, because it goes way beyond our field and I don't think it's really appreciated yet by traditional physiologists, how important the clockworks are for the regulation of fundamental normal activity throughout the entire body.
I couldn't agree with you more.
So that's an absolutely amazing contribution, and Mike Menaker deserves so much more credit than he's getting for this. Mike is an incredible mind. He's so thoughtful. He's a towering figure in our field, and I'm frankly appalled that he was not the first member of the more recent cadre of National Academy of Science members.
Well, going forward, what do you think are the challenges from your perspective?
I think from the clock's point of view, the challenge is to integrate what folks are trying to learn about the molecular biology of the clock with how that translates into a system. And I know they have a difficult time trying to extract clockworks out of a multiplicity of cells. So it's going to be probably an even more difficult time going from a multiplicity of cells, each with a molecular clock, to a system, an organized output that is communication via more typical action potentials moving down axons and so forth. I think that's going to be a big task for the future. And then, on the next level of integration, at least from my point of view, that's important, is bringing all these more peripheral regions in the brain into play to understand exactly how substances like NPY exert their effects on this collection of neurons in the SCN, and just as importantly, why they do. I mean, the NPY phenomenon is a big phenomenon. In terms of sheer magnitude of the phase response, but why it's needed even, or what it does to the ecology of the animal, in the context of the ecology of the animal. Nobody really knows. It's been a fun time. I wish NIH were looser with the purse strings.
It's been a fun time. I know, I feel we've gone from a time where when I first worked in this field, my colleagues would say, Martha, we like you, but why are you working in such a strange field? And now it's the meaning of life.
It is. And it's been very nice to be able to, at times, flip open science, an issue of science, and see, well, there's an article on circadian rhythms in there. And not only that, somebody's article on circadian rhythms made it into one of the winners of the year, as high points of in science for the entire year. It's very nice just to feel that you're part of that. When I got into the field was much narrower than it is right now, and the usual grant that was submitted, including all my own in the early 1970s, focused on two general topics. One is the role of the circadian clock in photoperiodic time measurement, and that is related to reproduction. And the other is on how the circadian clock worked and how that circadian clock sat within a system. And things have become so much more focused, and NIH study sections now require you to be so much more focused. A lot of that research effort has gone by the boards. There are extremely few people who actually studied the relationship between the circadian clock and reproductive function, for example, or any other function. It's on the clock itself. I'm not sure that's a good thing, because there's not really a place for it in the rest of NIH.
Don't you think it has to move in the other direction again, though? Because
at some
to
point,
get back to integrating all
I
the
sure hope so
systems.
at some point, but maybe not in our lifetimes.